Dr. Sabrina Paganoni. Welcome. She is a brilliant scientist and a compassionate clinician. She's also from the Healy Center in AMG at Mass General, where she is also the co-director of the Neurological Research Institute. Dr. Paganoni is also an associate professor at Harvard Med School, and she will be our first speaker today, giving an overview about the current clinical trial landscape. Following her talk, Dr. Paganoni will also moderate the first session. So, Dr. Paganoni, welcome and thank you.
Thank you, Jen, for the kind introduction, and thank you, Nicole and Chuck, for really setting the stage for this 3-day conversation, which is about progress, hope, and as you can see, there is an amazing community, over 100 people already, and many more will join, all here trying to really have a deep and insightful conversation about all the progress that we see in ALS and where we're heading. So it's an honor to be moderating the first session. The first session is about hot topics in clinical trials, and we have a fantastic panel of speakers. I will give an introduction about where we are in the ALS clinical trial landscape in 2024. And I will also highlight some of the current hot topics, and then each presenter will expand on them and really help us think through these important different topics and trial design options. As a moderator, one of my jobs is to keep us on time, especially in the virtual setting. So I believe that we will not have time for questions after each individual talk. However, we have a wonderful panel at the end of the morning session where we can really dive deeper into all the questions that you have. So please hold your questions and bring them back when we come together as a panel with all the speakers later in the day. And we'll have, I'm sure, a rich conversation about the clinical trial designs and hot topics in clinical trials. So I'll start by sharing my screen to provide the introduction and sort of set the stage for this first session. So where are we in the clinical trial landscape? So first of all, whenever we see somebody with ALS in the clinic, we see a process that started many years prior. So to the left, you see that everyone who develops ALS may have some mechanisms of disease that happened or started even years before presenting to us in clinic. And that can be different from person to person. So some people may develop ALS because of a genetic change or DNA mutation. Other people may have a number of different mechanisms that may be the initial trigger or become activated at different times in the disease process. The challenge is that when I see somebody in the clinic, it's hard to tell which of these mechanisms are at play. So we need specialized tests or emerging techniques and biomarkers to try to figure this out. Then as the disease progresses, obviously different symptoms arise, such as muscle weakness or difficulty with breathing. And then we measure in trials sort of, the effects on the whole body in terms of scales measuring functional outcomes. Now, typically in trials, especially the early trials, the first drugs that were developed looking at survival, and then more recently measuring function using the ALS functional rating scale in the revised form, which is one of the acceptable measures for testing drugs in ALS. More recently, there's been a lot of focus on trying to test and evaluate earlier phenomena such as biomarkers of motor neuron dysfunction or other measures of muscle weakness. And there is a lot of interest, as you'll hear from other speakers in the panel, about optimizing more powerful, more sensitive measures to develop an ability to capture these earlier events. And then in early trials, it's important that we also measure the drug's activity on target engagement, on the actual target. If the drug is meant to change, for example, gene expression, or if a drug is meant to affect one of the pathways like neuroinflammation or oxidative stress or anything that may be active at that time, it's important that we measure the ability of that drug to engage the target. Now, the challenge is that sometimes when we go from left to right on this graph, even trials that have shown good target engagement may have difficulties or challenges along the way, because then the sources of variability as the disease progresses continue to increase. And so we need to really pay a lot of attention, and that's why I think it's a hot topic, a lot of attention on the methodology that we use to test drugs. Clinical trials are scientific experiments, and we need to make sure that we control the variables, that we fine-tune the methodology, as you'll hear from different speakers today. And we need to continue to develop new methods and learn from ongoing trials. So just a very brief overview. The last 30 years of ALS research have have allowed us to form a number of collaborative initiatives and trials. And I do want to emphasize the importance of all these collaborative consortia that we see not only in the US, but also in Europe, in Asia, Australia, and there's these great connections and networks. And we come together once a year for the International M&D Symposium every December. So it's coming up in Montreal very soon. And all of these different networks and consortia have created a wealth of big data resources, collaborative initiatives, whether they are about genetics or biomarkers. And this is very important. So one thing that I want to mention, especially for the patient community, is that while we don't always have new drugs on the market, but there is a lot that's going on in the background. There's really a richness in terms of the data, and now we are really at the tipping point, because with all this data that's been collected, I think we really can leverage all of that to make progress faster moving forward. So far, 4 drugs have been approved in the US, and going left to right, Riluzole was approved in the 90s based on survival, sort of the right part of the graph I showed earlier, looking at the very late stages, kind of late effects of the disease, which is whether the drug prolongs survival or not. The 2 drugs sort of to the right here, edaravone and pibiterso, were approved in the US based on functional measures. As you know, edaravone was approved in 2017. And pibiterso was approved in 2022, but it was withdrawn from the market recently in 2024. And the only reason I mention it here is from a trial methodology point of view, because again, we're going to talk about hot topics in trial design, even if the drug is no longer on the market. And then last but not least, on the right, you can see the person that was approved more recently for people with SOD1 associated ALS. Now, these drugs were approved based on very, very different trials. And that's, again, what I want to focus on today. Trial methodology has evolved significantly over time. So let's look at Riluzole. Riluzole was approved based on trials that happened in the 90s. The 2 trials had a combined sample size or number of participants that was over 1,000, with very long study durations. The first randomized control trial was 12 months. The second was 18 months. And the primary outcome, again, was survival. So we were looking at sort of the ultimate kind of hard endpoint in ALS. So does the drug help with survival or not? The cohort that was enrolled was actually broader than what's typically enrolled these days. So it was a population with definite or probable ALS that had to have disease duration of less than 5 years, which is very different from contemporary inclusion-exclusion criteria for trials. Now, edaravone and pibiterso were approved more recently based on smaller trials, shorter trials that had a functional outcome as measured by the ALS-FRSR. And these were performed in very specific cohorts. You can see them on the slide, but essentially people with either definite or probable ALS in the case of edaravone within 2 years from symptom onset, and definite only ALS within 18 months from symptom onset in the case of pibiterso. So those were the designs of the trials that led to the approval. And again, then we'll talk about sort of the issue with confirmatory trials, and obviously pibiterso was removed from the market when the confirmatory trial had a negative primary endpoint. And then more recently, tofersen was approved under the accelerated approval pathway, which is very different from the full pathway per FDA regulations that was followed by the other drugs. And tofersen, again, was approved based also on a very specific, obviously targeted population of people with SOD1 ALS. And it was approved based on a randomized controlled trial, followed by an open label extension where the primary outcome was function, but the accelerated approval was supported by the biomarker results, specifically reduction in NFL levels. So when you look at kind of how the trial design has evolved over the last few years since the 90s, we can capture some emerging themes. So first of all, obviously, as I just described, eligibility criteria have been refined, and now they are designed to increase statistical power, trying to have smaller trials, more targeted trials, for example, in the case of tofersen, obviously with a genetic target. And then there's been a shift from focus on survival to focus on functions or biomarkers, because they can lead to a readout in a shorter time frame, or in the case of neurofilament light levels, they can be used to leverage the accelerated approval pathway at the FDA. And also in the US, there's been a few drugs that have been approved using single pivotal trials. However, there is the need for confirmatory trials. And then obviously, every time a new drug is approved on the market, it will affect the conduct of the future trials, because trials need to account for new standard of care, which can introduce some complexities. Now, so let's talk about a few strategies that were used by recent trials, recent approvals, and then we can discuss sort of pros and cons, and the next speakers will also dive deeper into some of these topics. But if you look at the most recent approvals, cohort enrichment has been used to increase statistical power. So obviously on the left, cohort enrichment can be done based on a biological mechanism. So obviously in the case of tofersen, the biological mechanism was having the SOD1 mutation. And so those were the people that were, they could plausibly respond to the treatment. Other trials have used mechanism-based selection, for example, people with certain biomarkers of inflammation or other biomarkers that suggested that the particular subgroup has a particular mechanism active at that particular time. In the case of edaravone and pibiterso, the trial design that was used was to actually create a homogeneous group of participants based on eligibility criteria using simulations and modeling on historical trial data. The goal here was to enroll participants with a similar, relatively homogeneous estimated rate of progression. So we know that on average, people with ALS progress by 1 point per month on the ALS-FRSR, which is this functional rating scale that we discussed. But then the progression can be very variable when you look at individual participants. And so the modeling was done, again, to narrow down the eligibility criteria to focus on that relatively homogeneous population. Obviously, when you do that, other questions arise, such as, are these results generalizable to the entire population? And also, how do you plan confirmatory studies? For example, if you go from phase 2 to phase 3, are you going to use the same criteria, or are you going to test the same drug that had a positive signal in a homogeneous population? Are you going to test it in a larger, broader population? So again, these kind of choices do impact, I think, the end results. The other contemporary trend has been to focus on earlier patients, because we think, and it's reasonable to assume, that if you target earlier patients, they're more likely to respond. As I said earlier, the older trials, the riluzole trials, actually had broader criteria for inclusion, meaning that people were included when their disease was ongoing for up to 5 years, which is very different from contemporary trials where we limit inclusion to, let's say, 2 years from onset. That has implication for survival analysis. If you look at the graphs of the riluzole studies, the impact on survival of riluzole was measured at the time when the population enrolled in the trial had already reached median survival, meaning they had basically 50% of participants already had survival events happen, and that gives you statistical power to see a difference between active compared to placebo. But in contemporary trials, actually mortality is much lower, even at 12 months. So modern trials, if you target early populations, are going to be underpowered to measure survival at 12 months. And this has implications for trial design. So just to kind of summarize all of this and just to set the stage for the next few speakers, hot topics currently are, what's the best target population? Every time we design a trial, we need to think about this. What's the best target population? What's the best duration of treatment? Should we use the usual traditional outcome measures, or should we develop and leverage more innovative new outcome measures? How do we move from phase 2 to phase 3, and how do we de-risk the program? And what's regulatory path should one use? The traditional two-trial approval route for full approval, or can we try to have a single pivotal trial, or maybe go for accelerated approval? All of these decisions impact the way trials are designed and conducted. And I will say that what I think that the main message that I want to convey is that I strongly believe that we should learn from data. And so data from previous trials should be made available to leverage, should be made available so that it can be leveraged to make data-driven decisions about future trial design. And the next speakers will talk a little bit about this and the richness of the data that we already have. Hopefully other ongoing studies will continue to contribute to these large data sets so that we can make evidence-based decisions in terms of designing new trials. So the good news is that we have a large number of trials. This is just a snapshot of trials that were available and enrolling in the US, sort of in the late stage, so phase 2 to phase 3. And if you go from, let's say, 20 years ago until, recently, you see an exponential increase. And I think this should be a reason for hope for the patient community, because it's amazing to see how many more new trials we have. We have more ALS trials now than ever before. And so this creates a great learning opportunity for two reasons. Number one, we can test more drugs. Obviously, that's great. But then also we can learn from our collective experience to continue to adapt and make trials better. And before I conclude, I do want to share, our personal experience, our experience at the Healy Center in terms of trying to leverage trials to learn and contribute to the field. And so I'll just spend just a couple of minutes telling you where we are with the Healy ALS platform trial. Again, not so much in terms of the results of the trial, but more in terms of trial methodology and opportunity for collaboration and growth. So we established the Healy ALS platform trial, launched that in 2020 as a very broad and robust academia industry partnership for efficient drug development in collaboration with many patient foundations. And this was designed in collaboration with many in the community to meet the needs of the community by reducing placebo, accelerating the timelines for testing, making more efficient use of resources, and also moving the most promising drugs forward from phase 2 to phase 3. And the basic principle of trial design here is that we want each regimen, and by regimen we mean testing one drug, we want each regimen to provide an efficient go no go decision to inform the next steps of drug development. So should this drug move forward to phase 3 or not? And that's kind of the big question here. And we do so by enrolling participants in this shared infrastructure where every regimen is fairly similar to the others in terms of having sort of an initial placebo control period where we test functional outcomes and survival, followed by an active treatment extension, where we and in both periods of the trial, we do deep genotyping, deep phenotyping. We collect a number of measures that can be a great learning opportunity again for the particular drug being tested, but also for the field. We launched in 2020 under a common protocol, so we have a very robust central infrastructure that has enrolled about 1,400 participants so far and continues to grow. 7 drugs so far, and we're very grateful for many partners, industry partners that have come together and under the same roof, under the same protocol to again develop drugs more efficiently and contribute to the field. And some drugs are moving forward to phase 3. Others, we don't have the results yet. That's kind of a snapshot of where we are with the different regimens. And for the most recent regimens, F and G, we'll have the results over the next few weeks. So the trial is also a unique opportunity to advance science and move biomarkers and new outcomes measures forward. So this is a list of the biomarkers and measures that we take from participants, including, again, genome sequencing, neurofilament levels, a variety of biomarkers, digital biomarkers, and we are also moving forward. We're going to be developing induced pluripotent stem cells from participants. And all of this, again, is an opportunity to learn for the field. We are actually actually adapting the trial based on our initial learnings. So we originally enrolled under the first 7 drugs under the same common protocol, and now we're adapting based on the experience on the first few regimens by increasing the length of the randomized controlled trial from 6 to 9 months and targeting a slightly earlier population within 2 years from onset. And we're also, again, fine-tuning the biomarkers and the opportunities that we have for learning. We are also retiring the data from the first few regimens that will no longer be used for analysis, and all of this data will be ultimately deposited in PROACT and will be available, again, for collaborative work with investigators from the placebo arms. And this, again, is just an example of how I think we should think about trials, where, when we complete trials, especially the placebo arms, it's important to think about, can that rich data contribute to the field? If it's no longer being used, I think it's to everyone's advantage to have that opportunity for collaboration to advance the field forward. We're sharing our experience. We have 3 papers published in case you're interested, and many more under review, and hopefully will be published soon. So we definitely are committed to sharing our methodology and our experience, because we are using that to adapt our own trial, but also hopefully can help others as they develop their own studies. So to conclude, I think it's really important to think that we are at the time in ALS where we have more drugs and more products in development than ever before. We have an extremely active, engaged, and collaborative community with robust partnerships, and so lots of opportunities to learn, adapt, and make data-driven decisions. And so with that, I hope that I kind of set the stage for our fantastic panel of speakers who will speak after me now. And again, please hold your questions. We'll have a great panel discussion at the end, and rich discussion and conversation about key topics in trial design in ALS. And